everyone, my name is Matthew Hufford and I am a research coordinator for the College of Medicine at the University of Arizona, and today I'll be talking very briefly about how a Brightfield microscope can be used for cell culture. So for context for the sort of research I'm doing, I'm working on p-selectin glycoprotein ligand 1 and a couple of different variants, SNP variants, and PSGL1 is this green homodimer right here. It is involved in the first stage in inflammation, in which it binds to p-selectin on the endothelium and initiates leukocytes transmigrating into tissue from the vascular lumen. PSGL1 is coded for by cell PLG, and it's the receptor for stimulated T lymphocytes, and p-selectin is on all white blood cells. It is a homodimer, and as I said, is important in inflammation and the immune response. So here's a diagram of an inverted microscope with the light source at the top and the condenser below that. And then there is the stage where light will be diffracted and refracted by the sample and the lenses through which one may view the sample. And here's a different diagram of an inverted microscope. So here is a video showing me looking at these are leukocytes, and this was the day before yesterday, and I was checking, I think it was yesterday actually, I was checking to see the confluence and whether I needed to split the flask, and as you can see that is a very full flask. That is 4x objective I believe, so that should be a 40 overall magnification. That'll be the 20, so this will be 200 fold, and it's difficult to record through the lens, but now I'm moving the stage around to get the leukocytes into focus. So I'm checking to see if there's any contamination, as well as to see the overall number of cells there, and to make sure that the nuclei are bright and healthy looking. You can see some of them are dividing. So yeah, I diluted this about 10 after I took this video. This is just a wild type, not the mutant mutants that we're studying. And to the right of this microscope is a fluorescence microscope that, never mind. And this is a flask that I thawed yesterday of endothelial cells, because I was going to run an assay, or I'm going to run an assay next week. And normally endothelial cells adhere to the plastic of the flask, but that takes time, and since I just took these out of liquid nitrogen, I'm again looking to see if there's contamination, and to see if the cells look like they're alive, and since they haven't adhered yet, they'll be floating around. But this is... Okay. So it's important to make sure that cells are growing properly, and there is no contamination. And there they are, still sort of shocked from having been frozen, but after a couple days they'll adhere and spread out and look like human pulmonary artery endothelial cells, which don't normally look that round. Yep, so that's all I got.